Virus, an Introduction, written by Surajit Dutta. B.Sc, Honours in Zoology, University of Calcutta, successfully completed in 2011. Ex-student of M.Sc, in Zoology, Vidyasagar University, Directorate of Distance Education, successfully completed first year in 2014. What is a virus? A virus is usually a very small, disease-causing agent, consisting of genetic material, single or double-stranded, DNA, or RNA, with a protein capsid. The capsid here denotes the outer shell of viruses. Some viruses may also have a second layer of lipid envelope. Some scientists claim that viruses are a state of genetic material, having complete freedom from cellular machinery. Some viruses possess single-stranded DNA, as genetic material, which is not found in any other living organisms. Figure. Examples of viruses, source of figure, courtesy, National Human Genome Research Institute, please visit the web link provided here. Why are viruses called obligate intracellular parasites? Viruses are obligate intracellular parasites because they cannot thrive or reproduce themselves without their host cell. Intracellular means viruses do live inside their host cells. Obligate here denotes the word compulsory. Recently discovered giant viruses also remain dependent on their host cell for reproduction purposes. How were viruses discovered? Viruses were discovered when Dmitry Ivanovsky conducted an experiment on an extracted disease-causing agent from a tobacco plant, which was later filtered through a bacteria filter that traps bacteria. When this extract was applied to another tobacco plant, it produced the same symptoms as the one from which it was extracted. It was soon identified as a disease-causing agent, not a bacteria, but something much smaller than bacteria. Later, Martinus Beijerinck named it virus, which literally means poison. Who discovered viruses and in which year? In 1892, Dmitry Ivanovsky, sometimes written as Ivanovsky, discovered viruses. Why were viruses called filterable viruses? Viruses were discovered through a bacteria filter, which traps bacteria and allows smaller particles to pass through. Therefore, they were called filterable viruses. Which virus defies the definition of virus as a filterable disease-causing agent? Giant viruses defies the definition of viruses as a filterable disease-causing agent, which literally means they can pass through a bacteria filter. Viruses were discovered in 1892 by a bacteria filter, which traps most bacteria and allows much smaller particles, like viruses, to pass through. However, after the discovery of giant viruses, this old definition of viruses, as very small disease-causing agents that can pass through a bacteria filter, was discarded. What are giant viruses? Giant viruses are a kind of virus, usually larger than most bacteria, containing both DNA and RNA, enzymes, and proteins involved in their metabolism. Photograph of mimivirus, a giant virus with two satellite Sputnik virophages. Denoted by arrows in the picture. Source: Wikipedia. Giant viruses were considered, for a long time, to be prokaryotes or bacteria. Which feature helps to identify them as viruses? Due to giant viruses having large size and complex structure, they were considered bacteria for a long time. Scientists arrived at the conclusion that giant viruses are truly viruses after studying their outer shape, which was an icosahedron. No other living form shows such a unique shape except viruses. Such a shape led scientists to conclude that they are viruses. Describe host specificity in viruses. According to the escaped gene hypothesis, viruses were part of the genetic material of their host cell, which escaped from their host cell and later developed the ability to jump from cell to cell. The similarity between the genetic material of viruses and host cells indicates their specificity to a particular type of host cell. During virus reproduction, the host cell membrane forms the outer shell of viruses. The proteins of the outer shell bind specifically to receptor proteins, located on a particular type of host cell. For example, polio viruses attack only nerve cells, hepatitis viruses attack only liver cells. Which bacteria possess virus-like features? Rickettsia, Mycoplasma, and Chlamydia are bacteria that can pass through bacterial filters. These bacteria are intermediate forms between bacteria and viruses. Why did scientists consider viruses as non-living assembly of nucleic acid and protein molecules? 1. Viruses are non-living assembly of molecules, since they do not grow, reproduce, or show metabolism outside their host. 2. Viruses can be crystallized, like any other protein, carbohydrate, non-living molecules. 3. Viruses disintegrate inside their host cell before using the host cell's cellular machinery. No other living organism shows such a feature of disintegration and also retains infectivity. Which properties of viruses are considered to be like any other living organisms? 1. Like any other living organism, they reproduce their copies when they are inside a host cell. Self-reproduction is a crucial property of living organisms. 2. They contain genetic material, like any other living organism. 3. They share the same universal genetic code with other living organisms. Why are viruses called nature's genetic engineers? Viruses create more copies of themselves by inserting DNA or RNA into host DNA. During the reproduction of viruses, they insert new genetic material from other organisms, which was accidentally collected when they were produced from other host cells. As a result, some traces of DNA can be found in humans from other organisms. Some scientists claim that human DNA possesses more viral DNA than human DNA. The result is that evolution is accelerated by viruses by introducing new genetic material from other organisms. This proves that viruses are nature's genetic engineers. How do we protect ourselves from viruses? 1. Keeping distance from people showing signs of having any disease. 2. Avoiding large, crowded areas. 3. Using a mask for outdoor activity. 4. Use soap to wash your hands thoroughly at 2 to 3 hour intervals. Soaps dissolve the lipid coating of viruses, which kills them. 5. Avoiding public toilets or washrooms. 6. Use a squatting-type toilet seat to avoid germs. Skin contact with toilet seats can sometimes spread contagious diseases. 7. Use hand sanitizer when soaps are not available. 8. Handling patients with masks and proper gloves is useful for avoiding contagious diseases. 9. Use precautions when handling laboratory testing samples, like blood, urine, etc. 10. Wash your hands with soap thoroughly after removing the gloves. 11. Proper vaccination prevents many diseases. 12. Constant self-education about viruses and pathogens is the most important key protection against any disease. Explain various hypotheses on the origin of viruses. Viruses are small disease-causing agents that show a state between the living and non-living. There are 3 hypotheses that explain the origin of viruses. A. Escaped gene hypothesis. According to this hypothesis, viruses are part of the genetic material of their host cells, which also reflects their specificity to particular host cells. The evidences which supports this hypothesis are: 1. Viruses have genetic material similar to the host cell genetic material, which also reflects their specificity to a particular host cell type. 2. Transposable elements, or mobile genetic elements, that show some independence from the entire genetic material of host cells. B. Degenerate life form hypothesis. According to this hypothesis, viruses are degenerated cells. The evidences that support this hypothesis are: 1. Rickettsia and chlamydia are bacteria that, like viruses, are obligate intracellular parasites, which means they live inside their host cells and cannot reproduce without host cells. Like viruses, these bacteria are much smaller than most other bacteria known. 2. Giant viruses have some cellular components, which indicate that these viruses evolved from more complex cellular ancestors. C. Viruses were the first form of life hypothesis. According to this hypothesis, viruses are the first form of life that connects the living and non-living states. This hypothesis is not supported because viruses have a sophisticated reproduction process within their host cell. They are totally parasitic in nature and cannot reproduce without their host cell. How are new viruses emerging so rapidly? Virus evolved rapidly due to various causes, which are explained as follows. 1. Defective synthesis of genetic material, which is common among viruses. Defective viral DNA, or RNA polymerases, synthesized during the reproduction of viruses, contribute to unique genetic material. Such polymerases are unable to detect errors in the genetic material. This type of genetic material has numerous new mutations, or small changes that can create a new kind of virus. Such viruses are able to infect other kinds of host cells. Such viruses express a new kind of protein outside their outer protein shell, which matches the viral receptor protein on a completely different host cell. They can now infect a new kind of host. 2. Genetic mixing in the host cell, which is attacked by multiple viruses. Host cells infected with 2, or 3, or a number of different strains of the same virus, sometimes produce a new kind of virus strain that contains new parts of genetic material from parents. Such mixing of genetic material produces a new kind of virus strain, able to invade host defenses more easily. 3. Environmental changes to viruses. The environment of viruses is drastically changing, because human curiosity has led them to new places where new viruses are coming in contact with humans. Such conditions were created because forests were destroyed to build houses, new roads and highways. Their natural habitat, where they live, is constantly attacked by human urbanization. 4. Climate changes. Viruses are now spreading to other parts of the world, because their reservoir hosts can thrive in a new environment due to global warming. 5. Arms race hypothesis. According to this hypothesis, viruses are under evolutionary pressure to fight back host defenses. Hosts are evolving new viral receptor proteins that inhibit viruses from entering host cells. Some viruses outside their protein shell use host molecules to protect themselves from the host immune system. This constant arms race with hosts leads viruses to evolve rapidly. 6. Evolutionary selection pressure. Modern medicine creates a new pressure on viruses to evolve rapidly. From natural selection theory, we know that varieties or variations exist in nature. Some people are tall, others are dwarfs. Some have immunity to certain diseases. Some people are white, while others are black, brown, etc. In a population of viruses, there are some viruses that are accidentally immune to antiviral medicine. Such immunity to an antiviral medicine is a type of variety that was not induced but existed from the beginning. Variety always exists in nature. Those viruses that survive the antiviral medicine will proliferate and produce a new population of viruses immune to that particular antiviral medicine. Such selection by antiviral medicine will produce a new kind of resistant viruses to that particular antiviral medicine. New medicine produced each year somehow acts as constant evolutionary pressure on the viruses. The example discussed above is producing new kinds of viruses with more immunity to specific antiviral medicine. Words to know. Some technical terms and their meanings are explained below. Polymerases are enzymatic proteins that assemble DNA or RNA polymers. Polymer denotes large molecules containing many subunits. Enzyme denotes a protein, or sometimes RNA, usually that accelerates a chemical reaction. Chromosome denotes large molecules containing DNA and histone proteins. Genes denotes a small section of DNA, or RNA, in viruses. Genetic material denotes DNA in most organisms. In the case of viruses, sometimes RNA. DNA denotes large molecules that contain the information of how proteins will be built. Some are structural, others functional proteins. This information is transferred from generation after generation. This information is also used during the baby's development in the mother's womb. This molecule is negatively charged. DNA profiling, or fingerprinting, is solely based on this property. DNA is located in the nucleus of cells. Some cellular organelles like mitochondria and chloroplasts have their own DNA molecules. RNA denotes a large molecule, similar to DNA but usually single-stranded. Double-stranded RNA exists in some viruses. RNA is located both in the nucleus and the cytoplasm of cells. Strand denotes a long string of nucleotides. Nucleotides are building blocks of DNA and RNA molecules. Virus strain denotes a subtype of the same species of virus. Mention the types of viruses according to host cells. 1. Animal viruses attack only animals, including humans. 2. Plant viruses attack only plants. 3. Bacteriophages or phages attack only bacteria. 4. Mycophages attack only fungi. 5. Cyanophages attack only blue-green algae or cyanobacteria. 6. Virophages attack only giant viruses. Photograph of bacteriophage T4. Source: Wikipedia. List differences between bacteria and viruses. 1. Bacteria are cellular, microscopic organisms. In contrast, viruses are non-cellular, ultramicroscopic agents. 2. Bacteria are usually large, and can be observed by the usual light microscope. Exception: Mycoplasma. In contrast, viruses are so tiny that the usual light microscope cannot be used. Exception: Giant viruses. 3. Bacteria can reproduce without any host. Exception: Rickettsia, chlamydia. In contrast, viruses cannot reproduce without their host. 4. Bacteria show properties of life. They can grow, reproduce, and show movement. In contrast, viruses show properties of both living and non-living. Viruses are inanimate objects when they are living outside their host cells. Only when they are inside their host cells, they disintegrate to reproduce more copies of themselves. Self-reproduction is a crucial property of living organisms. 5. Bacteria are usually beneficial organisms. A small number of bacteria are disease-causing agents. In contrast, viruses are usually disease-causing agents. Exception: some viruses, like phages, do play a role in controlling bacterial composition in the human digestive system. It helps the bacterial population to remain balanced. Such balanced microflora stops other pathogenic microbes from forming diseases. Sources of information. The information provided was collected from various books throughout my studies. Here are the current books and sources I have used to provide more updated and accurate information available at present. 1. Molecular Virology of Human Pathogenic Viruses, publication year 2017, author is Wang-Shik Ryu, see Chapter 1 and Chapter 21. 2. Advanced Biology Notes, publication year 1971, author is David Harrison, Ph.D., see pages 25 to 26. 3. By Britannica website, a short video on what is the difference between bacteria and viruses. Web link is provided here. 4. A short section on giant viruses, authors are Chantal Abergel and Jean-Michel from Current Biology magazine, available in PDF format, the web link is provided here. 5. A short section on the origins of viruses, author is David R. Wessner, Ph.D., website link provided here for more information. 6. A review article known as Chlamydial Metabolism Revisited, Interspecies Metabolic Variability and Developmental Stage-Specific Physiologic Activities. Website link is provided here for more information.